Today we're going to talk about biochemistry. Both inorganic and organic compounds must be talked about in order to do so completely. Biochemistry is the study of chemical composition and reactions of living matter. And substances that are inorganic do not contain carbon, whereas substances that are organic do contain carbon, specifically in combination with hydrogen. Examples of inorganic compounds include water, salts, and a lot of acids and bases, whereas carbs, fats, proteins, and nucleic acids, those are examples of organic compounds. Let's first look at water as an inorganic substance. So it's a very important polar molecule. Life wouldn't be possible without water because water allows for chemical reactions. All chemical reactions require water in some way. And water is an inorganic compound. So here you can see H2O. So it's two hydrogens covalently bonded to one oxygen. And it's polar, which means that it can also interact with other polar or other hydrophilic water-loving substances. And it makes up 60 to 80% of the volume of living cells. Here is just a quick list of some of the characteristics or properties that we're going to look at for water. High heat capacity, high heat of vaporization, the polar solvent properties, cohesion, surface tension and adhesion, and the density of ice. So first and foremost, water stabilizes temperature. This would be kind of a generalization for the properties of high heat of vaporization and high heat capacity. So water needs a great deal of energy or heat to affect its overall temperature. Water is also an excellent solvent. So it's something that can dissolve smaller things or solutes, things that are present in lesser amounts. Something that does the dissolving is known as the solvent. Water also has adhesive, cohesive, and surface tension properties, and this is mainly because of the hydrogen bonds that are demonstrated between separate or opposite water molecules. And these hydrogen bonds allow water to stick to other things, which is adhesion, water to stick to other water molecules, which is cohesion. And thanks to cohesion, we have surface tension in large bodies of water that allow organisms like a basilisk lizard and water strider to kind of skate or move across the surface of a body of water. So let's talk more about high heat capacity and high heat of vaporization. So high heat capacity is talking about the large amount of heat energy or energy in general required to increase the temperature of a body of water. And we need such a large amount of heat to produce even just a little bit of temperature change that the high heat capacity of water is indicative of its resistance to temperature change. High heat of vaporization has to do with the ability of water to absorb a lot of heat before evaporating, before going from a liquid to a gas. And that's how sweating is an efficient cooling system for the human body. Sweat contains a lot of water, and when that water evaporates, it's only doing so because it absorbed a lot of heat, and that evaporation process takes that heat with us, cooling the human body. Water is not truly a universal solvent, but it oftentimes gets that name. It can dissolve things like other polar molecules and anything that has a charge. Here's the definition for adhesion. It's an attractive force that holds molecules of different substances together. An example of this would be water sticking to the xylem or phloem channels in plants. So water can move from the roots to the stems or the branches of different types of plants. Water sticking to other water molecules is cohesion, an attractive force that holds molecules of the same substance together. And the density of ice, we want to talk about that because water is interesting when we look at its density. Solid water or ice is going to be less dense than liquid water. So solid water or ice floats above liquid water. Now, not everything can be dissolved in water. Again, that's why water is not truly a universal solvent, even though it oftentimes takes that moniker. Whereas water is polar, something like oil is nonpolar. And polar and nonpolar substances do not mix. And here we see oil and water, and they do not mix. We can see the fat droplets that are forming and resisting mixing or dissolving in water. So yes, water is an excellent solvent, and it can dissolve other things and yield a solution, but it's not universal, and it can only dissolve things that are positively or negatively charged, that has poles like itself as a polar molecule, and these things tend to also be hydrophilic or water-loving. Whereas anything that's hydrophobic or water-fearing is not going to mix with water. Again, it's polar, so non-polar hydrophobic substances like fats, oils, steroids, phospholipids, all of which are lipid or fat-based, are not going to be able to be dissolved in water. The reason that water is able to stabilize temperature or perform thermoregulation is not just because of a high heat capacity and high heat of vaporization. Those characteristics are possible because water has a high specific heat, which is just the statement that energy input must be high enough to break the hydrogen bonds between water molecules to excite those hydrogen molecules to break. And so if we don't have enough heat energy, we can't break those hydrogen bonds, and that's going to be a resistant nature to temperature change that water has that produces those characteristics of high heat capacity and high heat of vaporization. So high heat of vaporization, again, that's why water is a great cooling system, especially if we're looking at sweating as a cooling system for the human body, because water and sweat absorbs excess body heat and then cools us off as we evaporate, or as it evaporates. We talked briefly about ice having a lighter or being less dense than liquid water, and that's because the hydrogen bonds in ice are actually going to take on a lattice structure where the water molecules spread out more than they do when water is in its liquid state. So it's unusual that the solid form of a substance is less dense than its liquid form, but that happens with water. This is why icebergs and ice cubes float. And this actually allows aquatic life to persist below, and it prevents freezing of lower depths of large bodies of water, like in the ocean. So it's very insulating and protective for aquatic life. So here we see on the left, liquid water. Notice how close all of the water molecules are. So it's more dense than what we see on the right, which is the lattice structure of solid water or ice. So we can see how the structure of the water molecules and how they interact together is different on the left compared to the right. On the left again is the liquid. On the right again is the solid for water. And so we can see how the right picture, it makes sense that that's less dense or less crowded as the solid or ice form of water compared to the left, which is liquid. Here's just a better view of that lattice structure that makes solid water or ice less dense than the freely flowing molecules of liquid water. Let's talk a little bit more about the hydrogen bonds that allow for cohesion. Cohesion, which is water molecules sticking to other water molecules, or molecules of a single type sticking together, allows for surface tension. It allows for water flow, also droplet formation. So the surface of water does tend to resist being broken as long as something is not too heavy or much more dense than the liquid water itself. For example, you can take a needle, and if you lightly place it on top of water in a glass, you might see kind of a slight bend as the surface tension is pulled downward or is trying to resist the sinking of this needle. And if you agitate this cup too much, the needle will sink. But you can carefully suspend the needle on the surface of water and keep it from sinking, and this is because of surface tension. And surface tension is possible because of the cohesion between water molecules. Here is on the left, cohesion and adhesion being shown. So cohesion allows for the water molecules to stick together and help create water droplets. But the fact that the water droplets are sticking to the spider web is adhesion. And then on the right, we can see that cohesion is allowing for surface tension, and that allows for this basilisk lizard to run across the surface of this body of water. But we can't just leave adhesion alone and not talk about it in more detail as well. And again, this is when molecules of different types tend to stick together, and this is responsible for capillary action, or the movement of water into very narrow spaces. We see this movement of water into very narrow spaces if you were to put dye in water and then dip part of a paper towel in there. We also see this in action in plants, wherein water and water with dissolved nutrients in it sticks to the xylem and phloem, or canals in plants, to get materials from the roots to the shoots or stems and branches of a plant. So again, why is water so important? Not only does it allow for chemical reactions, it dissolves a lot of stuff, has all these interesting properties, but in large plants like trees, we need cohesion and adhesion to supply water and nutrients to these plants. This requires the use or utilization of hydrogen bonds, wherein the hydrogen bonds allow water to cohere to other water molecules, and then adhesion allows those water molecules to move up the vessels of the plant, and so that's utilizing capillary action and we're moving materials against gravity. So together, hydrogen bonds can be very strong, but before we continue on, let's kind of switch and talk about how water-based solutions can actually also be acidic, basic, or neutral. If you have just pure water, a small fraction of those water molecules, say in a beaker, do break apart into hydroxide and hydrogen ions. So you can see H2O on the left, no charges, but some of the water in a beaker of pure water will break down into OH negative, or a hydroxide ion, and H positive, or a hydrogen ion. And then some of the water in that same beaker would exist as formed H2O. And because when water breaks down, you'd have an equal amount of positive and negative charges, as long as we don't add extra H plus or hydrogen ions or extra OH negative or hydroxide ions, we would still have a neutral solution. But if we were to add a weak acid or a weak base, substances that could release either excess hydrogen ions or hydroxide ions, that could change that neutral pH or neutral concentration. And that change could create an acid or a base out of what was just a pure water-based solution. So here we see that pure water-based solution that sometimes has a water molecule H2O break down into hydroxide and hydrogen ions. And however many water molecules do that, we would maintain an equal number of negative and positive charge, and this would give us a neutral pH or concentration. But any solution that gets excess or more hydrogen ions than hydroxide ions would be considered acidic. We would call that thing an acid because acids release excess hydrogen ions, especially when dissolved in water. So you can see where water's coming into play. We get these freely floating or moving hydrogen ions when an acid is dissolved in water. A good example is hydrochloric acid, which ionizes or breaks down in water to create free hydrogen ions, which are what we're seeing in making this solution acidic, and then freely floating chloride ions. The freely floating hydrogen ions would exceed, in this example, the concentration of any present hydroxide ions, which is probably negligible or not as many as the hydrogen ions, again making this solution acidic. Examples of acidic solutions are lemon juice and vinegar. A base is the opposite of an acid. It's a solution with more hydroxide ions than hydrogen ions. Another term for something that is basic or a base is alkaline. And a good example of a base is sodium hydroxide. When sodium hydroxide is dissolved in water, we get a sodium or Na plus cation and a hydroxide or OH negative ion. And here we're going to have more hydroxide ions, and their concentration will exceed the amount of freely floating hydrogen ions in the solution. And so this makes this solution a base. Baking soda, different types of soap, things like bleach and ammonia are examples of alkaline substances or a base. Looking at the pH scale, we're interested in what does it measure. The pH scale actually measures the amount of freely floating hydrogen ions, or the H plus concentration. Something with a pH of 0 to 6 is acidic, a pH of 7 is neutral, and a pH of 8 to 14 is basic. Pure water would be an example of something that is neutral. So this is pure water, just H2O, with no minerals, vitamins, or anything like that added. And that would be because even though some H2O breaks down into charged particles, we'd always have the same amount of hydroxide and hydrogen ions in pure water. So here's an overview of kind of a basic pH scale. You can see on the left, we have the acidic examples. On the right, we have the basic or alkaline examples. And this is a pH scale, and the H in pH means we are measuring the amount of hydrogen ions. And there's more hydrogen ions in something acidic. So whenever something's acidic, we get a much lower pH being reported, something below 7. Whenever something is basic, we see a higher pH being reported, something above 7. And the pH scale is logarithmic. So something that has a pH of 1 is 10 times more acidic than something that has a pH of 2. So that brings us to buffers, and buffers generally exist as systems or a group of molecules that are trying to resist extreme changes or any change in pH. They're trying to help maintain a nearly constant pH of a solution. So they can tie up or kind of accept free hydrogen ions in response to something starting to become acidic, and that brings it back to a more neutral or basic pH. And they can also release extra hydrogen ions if something's starting to become more basic. And the release of free hydrogen ions makes something more acidic. So in response to something become more basic, that would produce a neutralizing effect. So essentially, in excess, or in the case of excess hydrogen ions, a buffer combines with those freely floating hydrogen ions. If we get excess hydroxide ions, a buffer will release freely floating hydrogen ions, and both of these responses are in order to help maintain, again, a nearly constant pH of a solution. In a lot of living organisms, it's important that the bloodstream, for example, stays at a kind of dynamic range or kind of homeostatic value for its pH. Changes one way or the other can upset the functioning of the bloodstream and the things that it transports. So if we did an example question, we could say, what kind of bond holds the atoms of a single water molecule together? The answer would be polar covalent. We're talking about the atoms that make up a water molecule, so we're looking at bonds that hold atoms together. But what about the kind of bond that forms between water molecules? For this one, the answer is hydrogen bonds, because the bond between complete water molecules gives us a lot of the characteristics of water, and those types of bonds are hydrogen. So make sure you read questions and you're looking at whether or not we're considering bonds between atoms or bonds between molecules. How about when you sweat, what property of water is functioning to cool you off? That's the fact that water stabilizes temperature. If you're looking for a more specific answer, you may have been looking for water has a high heat of vaporization, but that can be put with specific heat and the fact that water has a high heat capacity, which went under the broad category of water stabilizes temperature. So this is the best answer with what we're given. How about this question? Pure water is neutral in pH because it's because the concentration of hydroxide and hydrogen ions will be equal in pure water. But if we add an acid or base, we can disrupt that equal amount. And that brings us to the 4 major classes of biological macromolecules, sometimes called biological molecules. But these are large substances, hence the prefix macro. They include carbohydrates, lipids, proteins, and nucleic acids. And these are all organic molecules, so they contain carbon in combination with hydrogen. And they're all found in living organisms that also contain things like carbon, hydrogen, oxygen, and nitrogen, but in trace amounts or smaller amounts, sulfur and phosphorus. To create these large macromolecules, biosynthesis, or process that puts smaller subunits together to create larger molecules, occurs. Many of these macromolecules are very complex. They form sheets or chains or rings or helices, and this is possible because of carbon. Carbon is the foundation for many biological macromolecules because it has 4 electrons in its valence shell. Therefore, it can form 4 covalent bonds and create multiple types of organic molecules. So carbon is not only the foundation element for these molecules, it's also very versatile because it allows for the formation of those chains, sheets, rings, and helices because of the shapes it can take on. So when we talk about biological macromolecules, we have to mention monomers. Monomers are the name for the repeating subunits that come together to make or build macromolecules. Another name for the biological macromolecules are polymers because they're made up of many, which is the prefix poly, many of these repeating monomers. So mono as a prefix means one, poly means many. So if we put many of these one subunits together, we can get a polymer. Macromolecules are just chains of monomers, and they're formed by biosynthesis, specifically a type called dehydration synthesis, where we remove water from the bonds of 2 substances to put them together. And then if we add water, we can actually break apart the subunits of a macromolecule, and this process is called hydrolysis. So here's an overview of the biological macromolecules. We have carbs, proteins, nucleic acids, and lipids. Notice most of them have a subunit or a monomer. For carbohydrates, that's the monosaccharide. Proteins, it's the amino acid. And then for nucleic acids, it's the nucleotide. Lipids are not a true polymer, and they don't have a consistent subunit that we would call a monomer, but things that you would find that make up lipids are a lot of fatty acids and sometimes a sugar alcohol. Some examples of carbohydrates: glucose, fructose, sucrose. These are all smaller carbohydrates, and when they come together, they create polysaccharides like starch and glycogen, which are used for energy storage in plants and animals respectively, and cellulose and chitin, which are used for structural support in plants and then arthropods or insects respectively. Proteins have many functions, such as acting as enzymes or being a hormone or functioning in immune support by being part of antibodies. So insulin, oxytocin, hemoglobin, and keratin, those are some examples of enzymatic, hormone, and structurally important proteins. Something like insulin is involved in blood sugar regulation, whereas hemoglobin is an important, highly structured protein that allows for oxygen transport, and keratin is the structural component of hair. The main examples of nucleic acids are DNA and RNA. One type is messenger RNA, which communicates between the nucleus and the cytoplasm of a cell. Lipids, think of these as fats, and animal fats, things like butter and oil, those are great examples. But beeswax, triglycerides, steroids, like cholesterol, testosterone, and estrogen, these are also all examples. And lipids also are capable of performing energy storage or being a type of energy storage. And it's actually long-term energy storage in animals and plants, whereas carbohydrates were short-term. And structural components of cell membranes, that's talking about phospholipids. And phospholipids are going to be very unique, and we'll look at them briefly. And we'll talk more about them when we learn about the cell. So first, let's look closer at carbohydrates. So carbohydrates, you may think of them as carbs, and they have a lot of carbon within them bound to hydrogen, hence the name. Carbohydrates are carbon and hydrogen. And hydrate is also talking about the fact that water is needed for most of these molecules to exist. And most molecules found in a cell contain carbon, whether or not they're carbohydrates. And carbohydrates are the major structural and functional component of cells. Whether or not they're for structural support or energy depends on what type. So you see their main function is to provide energy to the body or structural support. If we're looking at a carbohydrate molecule, we can look at smaller ones or large ones. Small ones, or the monomer, is a monosaccharide. That's just one sugar. Disaccharides are two monosaccharides bound together. And a polysaccharide is many monosaccharides bound together. So the three subtypes, if we're looking at examples, glucose, galactose, and fructose, are examples of monosaccharides. Disaccharides includes things like lactose, maltose, and sucrose, and starch and glycogen, which are for energy storage in plants and animals, and cellulose and chitin, which are for structural support in plants and arthropods or insects. In order to create a disaccharide, we put two monosaccharides together, and we generally connect them where there is an H plus or a hydrogen ion on one side and an OH or a hydroxide ion on the other. A lot of monosaccharides are pentose or hexose sugars, meaning they have 5 or 6 carbons respectively. Glucose is one of the most common monosaccharides. It's the most common sugar in polysaccharides like starch and glycogen. You can see that glucose, fructose, and galactose are all hexose sugars and have 6 carbons, whereas ribose and deoxyribose that we see more in DNA and RNA are actually going to have 5 carbons and are pentose sugars. So here's the basic structure of a monosaccharide. So we can count the carbons: 1, 2, 3, 4, 5, 6. So since there are 6 carbons, this is a hexose monosaccharide. Again, disaccharides is if we put two of those together. Here are examples. We could put glucose and fructose together to get sucrose, or glucose and galactose together to get lactose. We put two glucose monosaccharides together, we get maltose. And those are the examples of table sugar, milk sugar, and malt sugar. So table sugar that you use is actually sucrose, or a disaccharide. So here you can see that if we remove water, so two hydrogens and an oxygen, via dehydration synthesis, the removal of water can create a bond, or we can put two monosaccharides together to produce a disaccharide. And polysaccharides are kind of the next in the chain of building or putting monosaccharides together. This is many sugars or many monomers put together to create large, complex carbohydrates, like starch and glycogen for energy and cellulose and chitin for structural support. Starch is what you find in plants for energy storage. For example, a potato contains starch. It's highly branched and contains many glucose monosaccharides. So here is its branch structure. You can see we have kind of these long branches that are connected. And we can see a detail of that. We can see that we have a lot of hexosugars because it is glucose molecules bound together. Glycogen is for energy storage, but in animals. For example, our glycogen is stored in our liver and skeletal muscles, and this is more branched than starch, but also made up of glucose. Cellulose is for structural support. You find it in plant cell walls, but it also can be utilized or broken down by some vertebrates that contain microbes that can break down cellulose. We don't have these microbes, so we can't break down cellulose, so this is what we consider fiber for our digestive system. So here's the detail of a cellulose molecule. You can see it is attracted to each other by hydrogen bonds, but we generally just get this cross-linking via these two branches, so it's much more elongated and not truly branched like starch and glycogen. Chitin is also for structural support, but in animals like beetles or sometimes spiders. It's also found in fungal cell walls, and it has some nitrogenous functional groups or nitrogens attached to these chains of glucose molecules. So looking at starch and glycogen, they have the most branch structure, whereas cellulose and chitin form chains, but they are all made up of monosaccharides linked together, so many sugars coming together to build these complex carbohydrates called polysaccharides. Next, let's move away from carbohydrates and look at proteins. So this is your protein quick guides. For example, they're a very abundant organic molecule. They are found in cells, in the bloodstream. They have various functions. Some of the main ones are acting as enzymes, which are catalysts that speed up chemical reactions, or hormones that allow the endocrine system to work. They can act to break or make new bonds, and they are made up of amino acids. The monomer for proteins is amino acids. And there's actually 20 different amino acids that we can find in the human body, and they differ based on their R group or their variable group, but otherwise they look pretty much the same. It's also really important what a protein looks like, or its structural organization is important. So its kind of globular shape or its structure, its shape, size, both of those things are going to be important to its function. And if you unwind or take apart a peptide, a polypeptide, or a protein, you can actually change its function or make it non-functional. So its shape is very specific to its function if we're talking about a protein. And if you change that structure, you can make a protein non-functional, and that process is called denaturation, and is usually accomplished by subjecting the protein to extreme temperature, pH, or chemical changes. So proteins have a variety of functions. We talked a little bit about enzymatic functions, so they're going to speed up chemical reactions. Plenty of structural support, keratin, collagen, those are both great examples. Nutritional albumin is not only found in the bloodstream, but also can be utilized to help make some of these macromolecules. Actin and myosin is found in skeletal muscle and provides the means for muscle contraction. Hormones like insulin and growth hormone regulate not only blood sugar, but metabolism in most cells. Antibodies are immune proteins. And even as kind of a negative example of a protein function, a lot of snake venom contains specialized proteins, and we consider these toxins, but they're actually very effective, and this shows you the wide range of functions or capabilities a protein can have. So structural, movement, defense, storage, signaling, and catalyzing reactions, or being an enzyme, those are the basic functions of proteins. Keratin is found in hair, collagen is found in your skin, actin and myosin in muscle cells, antibodies are proteins produced by plasma cells, as specialized immune cells, and venoms, even though they're not good for us, they're defense for other animals like snakes and spiders. Albumin, you may have heard of this being an egg white, actually provides nutrition and transport for chemicals around an embryo. Signaling insulin is a hormone that travels in the blood and talks to the pancreas. And proteins are enzymes. It's probably one of their most important functions. Amylase is an example of an enzyme that actually starts to break down carbohydrates. This actually begins in your oral cavity. So here are just some examples of proteins and how varied they can be or exist in nature. Hair, horns, and silk are variations of keratin and collagen proteins. Again, proteins are just polymers of amino acids. So the monomer for proteins is the amino acid. Smaller proteins are sometimes called polypeptides, but regardless, they're all made up of amino acids that have an amine group, a carboxylic acid group, and then a variable R group. So here's the basic structure for the monomer for proteins, a basic amino acid. Notice the central carbon attached to a hydrogen, and then on one side there's an amino group that contains nitrogen, and the other side is a carboxylic acid group that contains a carbon double bound to oxygen, and an OH or hydroxide functional group. This part at the bottom, or the hydrogen bound to carbon in the two yellow regions, is the same for all amino acids. Up here we have the variable group, or R group, and this is the only part of the amino acid that changes or is different among different amino acids. So for all of these amino acids, the bottom part is the same, and it's what's in green, or kind of this green-blue, that is different, or is slightly variable, and that's giving these amino acids different properties and chemical characteristics. So proteins are also really specific in how they fold. They have a three-dimensional structure, and that final three-dimensional structure is important for their function. Primary level is just the sequence of amino acids. We're basically seeing what order the amino acids are in. At the secondary level, that primary structure starts to take on shapes. Parts of the primary structures become a helix. Other parts become a beta sheet, or a pleated sheet kind of looks like a zigzag. When these secondary structures become globular or fold in on each other, we have entered the tertiary structure. This globular further folding is going to be determined by the environment and the type of amino acids that are nearby. And then the quaternary structure is when we have multiple tertiary structures linked together. So again, amino acid primary structure is the order of the amino acids. When this order starts to take on shapes like a helix or spiral, or a pleated sheet or zigzags, that's secondary structure. Tertiary structure is when the secondary structures fold together. So you can see we have a spiral here, and then a pleated sheet here, and they're becoming globular and folding together. And then a quaternary structure is when multiple tertiary subunits come together to create an even more complicated protein. And these four levels of protein structure can be observed in these illustrations, but it's also important to note that especially at the tertiary and quaternary level, we have a lot of kind of hydrophilic and hydrophobic interactions dictating how the protein looks. Some amino acid R groups are more hydrophilic and others are more hydrophobic, and they like to kind of stay away from each other, or the hydrophobic regions like to be in the center of a particular protein, and these interactions can make proteins amphipathic, meaning once they're folded, they have areas that are water-loving and areas that are water-fearing. Here's another view of that 4-scale structural support that we see in proteins. Primary, order of amino acids. Secondary, shapes given to the primary structure. Secondary structure becoming globular is tertiary. And for quaternary, we have multiple tertiary subunits. Here in this example, we have hemoglobin, which is actually made up of not 1 or 2, but actually 3 and 4 So 4 total polypeptides to create this quaternary structure. And secondary and tertiary structure determine a lot about the function of a protein, but that precise positioning of an amino acid is pivotal to its function. It determines what regions interact with what, and denatured proteins have had their tertiary and secondary structures disrupted, and if that's permanent, if only the primary structure is left intact, denaturation can cause functional loss of proteins. If you've ever cooked an egg, then you know you can't uncook the egg, and that's because you have denatured the albumin proteins in the egg white, and once they are denatured to a certain degree, they can no longer perform their function of being an embryo support system. Let's talk more about proteins that are enzymes. So enzymes are biological catalysts. They speed up chemical reactions. They speed up chemical reactions by binding to something specific. What enzymes bind to is called the substrate. When they bind to that substrate, they either build or break apart bonds. And a lot of enzymes end with the suffix ase, something like hydrolase, oxidase, catalase, or even amylase. And sometimes the name gives you an idea of what it breaks down. For example, lipases break down lipids. The basic steps in enzyme action are when a substrate binds to its active site, or the specific substance that binds to an enzyme binds to the area it recognizes. This creates an enzyme-substrate complex, sometimes called the ES complex, and this is when the catalyzation process begins, where the rearrangement of bonds or the breaking of bonds is going to occur, and some type of product will be released, whether it's putting two monomers together or breaking them apart. Enzymes catalyze reactions and help us build and break down biological molecules. Here's the basic mechanism of enzyme action with a different picture. So here we have an enzyme. It's going to take in substrates that fit the active site, forms an enzyme-substrate complex. Catalyzation occurs. Here we have dehydration synthesis, where water is removed to make a bond, and that newly formed peptide bond with its 2 amino acids will leave the enzyme, and the enzyme will be unchanged, and it can repeat this mechanism over and over again to help build a polymer. So the active site is just the area of an enzyme to which a specific substrate fits, whereas the substrate is a specific reactant that's acted on by the enzyme. So let's look at enzymes even closer. We'll do a quick mini lesson. And most of the chemical reactions within living systems are controlled by specialized proteins called enzymes. So don't forget that enzymes are a type of protein. Biological catalysts are common keywords when we're talking about enzymes because they speed up chemical reactions. And they're not used up in the process. So each one is also specific for a type of chemical reaction. So each enzyme is specific for the catalyzation of a different reaction. And cells need enzymes to create biological molecules. And we also utilize enzymes in the creation of genetic material in order to read it so that we can get information about building proteins. So enzymes are proteins, therefore enzymes are also made up of amino acids because amino acids are the monomer for proteins. And most enzymes contain 100 to 1,000 amino acids, but they can form very long chains, and just like the general information given for proteins, enzymes are going to take on a three-dimensional shape that is very structurally important to how they work. That's because enzymes are specific, and how they fold creates an active site to which only one type of substrate will bind. And so different types of enzymes have different shapes and functions because of their sequence of amino acids and then their secondary and tertiary structure, and that gives enzymes their very specific function. All right, let's look at enzymes in a little bit more detail still. All right, not only are enzymes specific, they're also pretty picky. They like certain environments. They're not only looking for a certain reactant or substrate, they like a certain pH or a temperature. Again, the substrate is the reactant that the enzyme acts upon, and the substrate is going to bind to the active site. And again, very specific shape for the active site and substrate so they fit together. That's why enzymes are specific, and their three-dimensional structure is important. Sometimes we say enzymes bind together via lock and key. And we can get an enzyme reactant complex when the key or the substrate fits in the lock or the active site. And then catalyzation happens and we get enzyme and product. And the way that enzymes speed up chemical reactions is by lowering the activation energy. If we don't have an enzyme, oftentimes the activation energy is much higher than it is with an enzyme. So you can see without an enzyme, the hill that the reactants must overcome in order to produce products is much higher than it would be with the enzyme. So with the enzyme, that activation energy, or the hill the reactants must overcome, is less. Here's just another view. We have the left without enzyme, the right with enzyme. You can see less activation energy is required if an enzyme is present. So we can see the progress of the reaction proceed much quicker with the enzyme than it would without. And the rate of enzyme catalyzed reactions can depend on a couple of things. I mentioned that enzymes are picky about pH and temperature and the amount of substrate, the amount of enzyme present. So these are all factors that affect the rate of a reaction: temperature, pH, enzyme concentration, and substrate concentration. We need enough enzyme and substrate. If you're out of substrate, the enzyme may be there, but it's nothing to bind to, or there is nothing for it to bind to. If you change temperature and pH, enzymes oftentimes unwind or become denatured, and they won't work as well. Now, if you slightly change temperature or pH, you might be able to speed up the action of that enzyme, but you have to be careful. So all enzymes work best at only one particular temperature and pH, and that would be its optimum temperature or its optimum pH. And different enzymes have different optimums, whether it's temperature or a pH value. We don't want to change it again too much because we could irreversibly disrupt the protein shape. And if we affect the shape, the enzyme might no longer work. And this would be a process called denaturation, or we would say that when an enzyme unwinds and becomes non-functional, it's denatured. So you can see normal on the left, too much heat or too much change in pH can cause the denaturation or unfolding of the enzyme, making it non-functional or not specific enough to bind to its substrate. You can also have inhibitors or activators. They bind to a different location on an enzyme called the allosteric site. Some of these activators are also known as cofactors or coenzymes, and you may have guessed that inhibitors are going to decrease enzyme activity, whereas activators will increase enzyme activity. To a certain extent, if you increase temperature, you can actually increase reaction rate. You don't want to do it too much and denature a protein or enzyme, however, because at a certain point, that temperature or pH gets too high, and we have an enzyme that can no longer bind to reactants. So terms you should know in relation to enzymes: substrate, active site, enzyme substrate complex. Review things like dehydration synthesis and hydrolysis as well. And then don't forget to make sure you know what denaturation is. And just a little bit more on enzymes. Like here is a quick glossary of things like what is activation energy, which is the energy needed to start a chemical reaction, or active site, the part of the enzyme in which the reactant molecule fits. Don't forget that enzymes are catalysts because they speed up chemical reactions, and a catalyst is any substance that changes the rate of a reaction without being used up itself. And enzymes are always ready to go after they've catalyzed a reaction. They're not used up or broken down during that chemical reaction they facilitate. A basic experiment that you could do at home is to get 3 test tubes or cups, label 1 hydrogen peroxide, 1 sugar water, and 1 water. Fill them all the same amount with what they are labeled as: water with sucrose mixed with water for sugar water, and some hydrogen peroxide. And then get some chicken liver and cut 3 little pieces of chicken liver and put them in each cup or test tube. You'll notice that the chicken liver you put in hydrogen peroxide will produce bubbles. These bubbles suggest a chemical reaction, and this chemical reaction is breaking down hydrogen peroxide into H2O and O2 because liver contains catalase, and that is a very important enzyme that breaks down radical chemicals like hydrogen peroxide. The liver actually needs to utilize these specialized enzymes to break down toxic substances that could cause the mutation of DNA or cell damage. So within our liver, we also have catalase, and we have a lot of catalase in many tissues, and it causes a chemical reaction that neutralizes peroxidases like hydrogen peroxide. So the enzyme catalase is neutralizing peroxidases, which are also known as reactive oxidating species because they can cause oxidative damage like cell damage and DNA mutation. And even though hydrogen peroxide is a byproduct of cell respiration, which is a process that produces ATP, hydrogen peroxide is a waste product that our cells have to deal with on a daily basis. So it has to be broken down to prevent any cell or oxidative damage, because we don't want to mess with the DNA or the cell membrane. So by itself, hydrogen peroxide can be destructive. So it's best to break it down into water and oxygen. So that means the bubbling you see when you put the liver in the hydrogen peroxide is actually bubbles filled with oxygen gas. And last but not least, let's talk about nucleic acids. So with nucleic acids, our best examples to give are DNA and RNA. DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. DNA contains the genetic material or blueprint for how to make a protein, and RNA uses that blueprint and actually directly is involved in protein synthesis. So nucleic acids made up of nucleotides as their monomer include DNA and RNA. DNA is double-stranded, whereas RNA is single-stranded, and they have similar bases. However, DNA has thymine, whereas RNA doesn't have thymine and has uracil in its place. If you're looking at the monomer of a nucleic acid, you want to look at nucleotides. And when nucleotides are broken down, they're broken down into these three components: a sugar, a phosphate group, and a nitrogenous base. The 5-carbon sugar is a pentose sugar like ribose or deoxyribose. Deoxyribose is found in DNA, and ribose is found in RNA. DNA with its double helix is more important to the blueprint or the working of cells, but without RNA as an intermediary or go-between molecule, we couldn't get that blueprint material from the nucleus to the proper cell machinery in the cytoplasm. Also notice the basic structure of DNA. The nitrogenous containing bases are in the center, and the sugar and phosphates make up the backbone or the sides. So the sugar phosphate backbone with the nitrogenous bases in the center is the basis for the structure of DNA. Also notice all the dotted lines. These dotted lines represent hydrogen bonds, which actually hold the two strands of DNA together. And remember, hydrogen bonds may be weak, but together they're very strong. So subunits of nucleic acids are the nucleotide. We talked briefly about what they are made up of: a nitrogen-containing base, sugar, and phosphate. So here is that structure. Here's just another image of it, so we can see another nitrogen-containing base. The phosphate group is centralized phosphorus connected to oxygens, and you have that pentose sugar or 5-carbon sugar. Functions of nucleic acids are critical energy carriers like ATP. We'll talk later about cellular respiration in this class. Subunits of nucleic acids are nucleotides, and those can have functions when they're broken down and we use their parts. And they also act as intracellular messengers. So ATP is our critical energy carrier. We can break apart ATP to produce energy or release energy that's stored in the chemical bonds. The intracellular messenger example is mRNA. So it communicates between DNA in the nucleus and the cell machinery in the cytoplasm. And then our last biological molecule to look at is lipids. Lipids are hydrophobic, and they don't mix with water, which is polar and hydrophilic. The main components of lipids are glycerol and fatty acids, but I can't say those are the monomers. Lipids are not a true polymer, so they can't have a true monomer. But the examples we're going to look at are fats, both saturated and unsaturated, phospholipids, and steroids and waxes. So saturated fatty acids are solid at room temperature, and unsaturated fatty acids are liquid at room temperature. They provide great protection and insulation, and in certain locations are stored for long-term energy. Phospholipids are found in the plasma membrane and are actually their major component. You can also find things like cholesterol in the cell membrane, and then waxes like beeswax and lanolin provide waterproofing to not only structures, but also we tend to find certain types of waxes on plants to prevent tissues from becoming waterlogged. So here are the basic structures for some lipids. Saturated fatty acids tend to be made up mostly of hydrocarbons, or hydrogen and carbon bound together. You take a saturated fatty acid and connect it to a sugar alcohol, and you do that 3 times, you can create a triglyceride. Sometimes triglyceride tails, or these hydrocarbons, are bent. That's called having a kink. And that would mean that the tails are unsaturated instead of saturated. Unsaturated fatty acids don't stick together as well because of the kink in the fatty acid tail. And that's why unsaturated fatty acids are liquid at room temperature, like an oil, whereas saturated fatty acids are solid because their tails fit very closely and can pack together. A phospholipid similar to a triglyceride, it has one less fatty acid tail and a phosphate group, and steroids are going to have a ring structure. An example of a steroid would be cholesterol, or the estrogen and testosterone hormones. So there's a variety of functions to lipids, like there is for a lot of our biological molecules. Like carbohydrates, they're for energy storage. Carbohydrates are for short-term energy storage, whereas lipids are for long-term energy storage. We talked about the waterproofing capabilities of waxes. And then they're the primary component of cellular membranes. This includes both steroids like cholesterol, but the primary component is actually phospholipids. And then hormones are derivatives of cholesterol. The main ones we're going to talk about that are lipids will be estrogen and testosterone as the examples, which help regulate the maturation of male and female sex characteristics. Another thing to talk about if we're talking about waxes and their use, some animals actually produce waxy substances that they rub on their fur to make them a little bit more hydrophobic or water resistant, and this is actually mainly found or seen in animals that are aquatic, such as the river otter. When you're talking about lipids, you can put all of them into these 3 categories: an oil, a fat, or a wax, phospholipids, or a steroid. Oils, fats, and waxes tend to be made up primarily of fatty acids. A lot of times they combine with sugar alcohols called glycerols, and a triglyceride is a great example. It contains 3 fatty acids and 1 sugar alcohol. So we see 3 yellow fatty acid tails. Here we have glycerol, the sugar alcohol. Via dehydration synthesis, they come together to create a triglyceride. Fats and oils are the most energy-rich of the types of lipids we're talking about. They have twice as many calories as a similar size or amount of carbohydrates and proteins. So fats should be 100% avoided, but we primarily want to avoid the ones that are produced by animals. We're more interested in things like unsaturated fats that are liquid at room temperature, whereas saturated fats are going to be solid at room temperature. They have no double bonds between the carbon atoms that make up their fatty acids, meaning they can be closely packed together, and molecules that can be closely packed together are more likely to be solid versus liquid. Oils are mainly made up of unsaturated fats. They're liquid at room temperature, and they do have kinks or double bonds between their carbon atoms. And this means that we can't pack as many closely together, and they're loosely packed and making them a liquid. So here we have a saturated fat, like butter, solid at room temperature, and you want to limit these to reduce your risk for heart conditions later on in life. And then it's healthier, not necessarily the most healthy. You want to consume nothing in excess as best as you can help it. But here we have an unsaturated oil. We can see all the kinks and bends in the fatty acid tails, and these are hard to pack together, so they tend to be liquid at room temperature. Wax is very similar to fats. Some are actually going to be highly saturated, so solid in outdoor temperatures. Very rarely are they unsaturated, but they're definitely water repellent, and they can prevent fur, feathers, plants from becoming waterlogged, and they're also useful for bees to create honeycomb, so they can provide some structural support. So here's bees and their honeycomb, and this is primarily made up of water-resistant wax. Phospholipids look a lot like triglycerides, but they only have 2 fatty acid tails, and they're bound also to a phosphate group. This gives them an interesting dynamic. They have a water-loving or hydrophilic head and a hydrophobic tail region. This means that they're amphipathic, or they have areas that are hydrophilic and hydrophobic. So the head is polar and hydrophilic, and the tails are hydrophobic or nonpolar. And finally, steroids take on a ring structure, and cholesterol is kind of the base example for the ring structure that we see in steroids. A ring-structured molecule is sometimes called aromatic, and these are vital components for nerve cell insulation, but mainly we're going to talk about them being used to synthesize other steroid hormones like estrogen and testosterone. So here you can see we have the base cholesterol. If you change it up a little bit, shorten these hydrocarbon chains that were attached, you would get steroid hormones. And both steroid hormones are able to help regulate the maturation of sex characteristics in females and males respectively. And here let's try a sample question. Which is not a type of lipid? The answer is peptide. So peptide is a small protein, but triglycerides, waxes, and oils, those are lipids. And here is a lecture activity that you can complete either during or after the lecture. This is going to be dropped off, so you may have had this during the entire lecture activity. If that's not how this worked out, you can access this lecture PowerPoint on Teams, or you can also get it. It was sent via email. If you can't find it or you need it, please email or contact me, and then complete this for homework if you did not do so during class.